All right, good afternoon. So we had an exciting morning. I really enjoyed all the talk. So we're starting our senior session in the next hour. So I'm excited to have Dr. Chang, one of our neuropathologists that we work very closely every week, every day almost. And for CNS, I would say this is a very hot area utilizing artificial intelligence or radiomics, a lot of studies. So I'm looking forward to hearing her talk on applications of AI in brain tumor diagnosis.
Great, yeah. So thanks, Dr. Zhang, for the introduction. So I'm Ji Chen, one of the neuropathologists here. Today, it's really my great pleasure to discuss with all of you applications of artificial intelligence in brain tumor diagnosis. Okay, so, all right. So now artificial intelligence is widely used in the field of neuro-oncology by neuroradiologists, oncologists, surgeons, and because I'm a neuropathologist, today I will focus on how pathologists are using AI for measurement of features, tumor classification, grading, by marker identification, and diagnostic assistance. So in particular, this afternoon, I will be focusing my talk on the newly discovered technique called methylation profiling. So if you are not very familiar with methylation profiling, we'll start with the cover page of the new WHO, because this cover image is really illustrated the central idea of the so-called integrated diagnosis. Because now for brain tumors, we have to incorporate the morphological features as well as molecular findings to give a integrated diagnosis. And for example, these three images here are illustrating the techniques that we are using, sequencing, fluorescence in situ hybridization, and the last image is actually methylation profiling. Okay, so this is an enlarged image of the cover image. I just directly copied and pasted from the WHO. So this is the t-SNE projection of methylation array profiles from ependymal tumors. t-SNE, the full name is written here. So this is actually a technique to visualize the methylation profiling data. So here we can see the different subtypes of ependymomas. For example, the supratentorial ependymomas are here, the posterior fossa ependymomas are here, and the spinal ependymomas are here. So they are really very clustered nicely and separately on this t-SNE plot. So besides subtyping of ependymomas, methylation profiling is actually also being integrated into the diagnostic criteria for some tumors. One example of that is high-grade astrocytoma with pilot features, also known as HGAP. So we can consider this tumor as what we used to call anaplastic pilocytic astrocytoma. So, and later after, study them by methylation profiling, people realized that those tumors actually clustered very nicely into one separate cluster, different from all the other known entities. So now in the WHO, the methylation profile of high-grade astrocytoma with pilot features is listed as an essential criterion for the diagnosis of HGAP, which means without doing methylation profiling, we cannot make this diagnosis. All right, so now I hope you understand that methylation profiling is really integrated into the new WHO classification system. So we'll dive deep into the technique itself. Nowadays, the most widely used technique is through the Infinum methylation epic bead chip microarray. So this technique can identify the methylation status of 850,000 CPG sites throughout the human genome. And so what they do that is after DNA is extracted from the tissue, the first would undergo bisulfite conversion. In this step, any methylated C will stay as a C, but any unmethylated C will be converted to a U. And then the product will be hybridized on this bead chip and then will be labeled by different fluorescent dyes and then scanned, and then the data will go through very complicated bioinformatic pipelines. And then the first group to use methylation profiling for brain tumor diagnosis was actually the DKFZ Heidelberg group. They published their data in 2018 in Nature. So what they did is they developed a machine learning-based tumor classifier, and they found that they will actually change the diagnosis in up to 12% of their cases. Okay, let's take a closer look at what they really did. So first, they analyzed the data from more than 2,000 samples and established this reference cohort. In this cohort, we can see there are 91 classes of different brain tumors. They roughly corresponding to the WHO entities. We can see the medulloblastomas are here, ATRT, GBMs are here, and then we have ependymomas and meningiomas. So after establishing that cohort, they use this reference cohort to classify their test samples through this machine learning random forest classifier. So basically this random forest classifier would give a raw score to each test sample, and then later the raw score will be transferred into a calibrated score. And then this calibrated score will representing the probability of one tumor really matching to a specific methylation class. So for example, if the score is greater than 0.9, it will be considered a high match with confidence. And then if the score is less than 0.1, it will be considered as no match. And then they use this classifier to classify another more than 1,000 cases. And then they compare the original pathological diagnosis and the report from the methylation profiling to see if there are any discrepancies. And then indeed, they found that even in 76% of the cases, methylation profiling was able to confirm the original pathological diagnosis. However, in about 12% of cases, methylation profiling actually was able to establish a new diagnosis. All right, so this is the figure showing the 12% of cases with discrepant results. For example, in the group with a pathological diagnosis of diffuse astrocytoma IDH wild type, actually many tumors were upgraded to GBM grade 4. And then similarly, in the group with a pathological diagnosis of GBM IDH wild type, many tumors were downgraded to, let's say, PXA, ependymomas, and even low-grade gliomas like pilocytic astrocytoma. So we would imagine that these changes would have altered patient management. All right, now the methylation classifier, the DKFZ classifiers are publicly available on this website. So if you submit your methylation profiling data to their website, within a few minutes, you will get a report with the methylation class identification, calibrated scores. You can also get the copy number variation data as well as MG, MT promoter methylation results. And then now in the US, the NIH is becoming the largest reference center for methylation profiling. They also published their data recently, and based on their results, looks like in about 27% of cases, methylation profiling was able to give a new diagnosis. So now the NIH methylation data are also publicly available on this website. So if you click one dot, you will know which methylation class that tumor matched to, and then you will also get the copy number variation data. So I hope now you understand that methylation profiling is very helpful. It can help us accurately classify brain tumors into specific molecular tumor groups. Actually, methylation profiling can also help us identify new tumor types. I'll start with a case that we had at UNMC. So this is a 63-year-old male with a very large heterogeneously enhancing mass along the anterior inferior frontal lobe. When we got the slides, we saw very pleomorphic cells with many mitotic figures and areas of necrosis. In some areas, though, there are some interesting features, and this tumor cell seems to be surrounding the vessels forming the pseudopapillary architecture. And then GFAP was diffusely positive, so we know that we were dealing with a high-grade glioma. In this patient population, of course, the first thing we would think the top differential would be, could this be a GBM? However, when we did neurofilament thing actually only highlight the axons surrounding the tumor, not within the tumor. This was kind of argue against the diagnosis of GBM because we would consider GBM as an infiltrating glioma rather than a well-circumscribed glioma. So because of these conflicting features, we decided to do additional molecular testing. We did fluorescence in situ hybridization, which revealed polysomy 7, monosomy 10, and homozygous deletion of CDKN2A. And then we further did NGS with our new POP300 panel, in-house panel, which revealed mutations in RB1 and TP53. And still, we were not quite confident to directly call it GBM, and then we decided to send it to NIH for methylation. And then it turns out that this tumor actually matched to a new entity called HPAP. And then we also got the copy number variation results showing, again, polysomy 7 here, monosomy 10 here. It additionally showed monosomy 13. And then, yeah, if you don't know what HPAP is, yeah, it refers to high-grade glioma with pleomorphic and pseudopapillary features. This is a newly discovered entity by methylation profiling by the NIH group. So those tumors are considered as circumscribed glioma with frequent TP53 mutations and the recurrent monosomy 13. And then by morphology, they also show very pleomorphic cells. They also have these pseudopapillary features. These features were identical to what we had in our case. So, and then you may ask, well, then what's the significance? Well, what does it matter whether I call it GBM or HPAP? Well, if you look at the survival data, you would see that HPAP actually have a much better prognosis compared to GBM. All right, so methylation profiling is definitely very helpful. It also has several limitations. First, the array-based methylation profiling technique is very expensive because first, the instruments are expensive. It will also need to have expertise to handle those complicated bioinformatic pipelines. Currently, unfortunately, there's no specific CPT code for billing. Therefore, now the array-based methylation profiling is only available in a few large referral centers. We, UNMC, as the largest medical center in Nebraska, cannot do methylation profiling by ourselves. Therefore, now methylation profiling always needs a long turnaround time. It usually takes up to weeks or even months. Also, methylation profiling requires high tumor concentration. That's one reason that now actually only about 20 to 30% of cases cannot be classified confidently. So just, some things about future directions. Of course, we need to improve the classifier, and this has been an ongoing effort. For example, on the website, we can see that the DKFC group has been constantly updating their brain classifier. The first version was 11B2. Now the most updated version was 12.8. They have also been trying to develop other classifiers for sarcoma and meningioma. So this is an ongoing effort. Okay, the other thing is, since the array-based methylation profiling is very expensive and not widely available, people have been looking into other alternatives. One of that is nanopore sequencing. The advantage of nanopore sequencing is it can directly detect methylated sites without biosulfite conversion. This is because the methylated bases will alter the ionic current, which can be read out directly. Therefore, nanopore sequencing can provide rapid or almost real-time data generation with relatively low setup cost. However, the disadvantages of nanopore sequencing is that nanopore sequencing can only generate relatively sparse methylation profiles. And another very big drawback is that it requires a separate algorithm or classifier, which means the widely used DKFC classifier cannot be directly used for nanopore sequencing. Regardless, non-per sequencing seems to be a promising technique, and people have been using it for fast tumor classification during surgery, as well as trying to classify tumor from cell-free DNA from cerebral spinal fluid. All right, this is the summary slide. So I hope now you understand that whole genome DNA methylation profiling is a very robust technique that utilizes machine learning to improve diagnostic accuracy and to identify new tumor types. However, DNA methylation array is very expensive, time-consuming, and not widely usable. On the other hand, non-per sequencing may potentially perform very rapid methylation-based classification in a relatively low throughput setting. That's all I have today. Thank you very much.
All right, thank you, Dr. Chen. So we'll hold on all the questions to the end of the QA session as always. So our next speaker is, again, very familiar to our department. So Dr. Nicole Shankar has been, I would say, one of the most important neuro-oncologists in this region, and we definitely share a lot of patients. So besides being a busy physician, she runs quite a busy lab as well with NIH R1 funding, and congratulations on promotion to full professorship. So today she's going to talk about repurposing FDA-approved drugs for glioma treatment, which shares quite a common interest of mine. So look forward.
Thanks, Kevin. So I'm going to talk a little bit about repurposing FDA-approved drugs for glioma therapy. So, one of the big challenges that we have is that, we all are very familiar that quite often it takes more than a decade to get a drug from lab to actually into a patient in any way, shape, or form. So one attempt to try to speed this up, also using bioinformatics, is looking at, FDA approved drugs, ones that are already approved and using them for other things. So there are my disclosures. We're going to talk first a little background about glioblastoma for everybody, and then specifically move on to connectivity mapping, and then the 3 different projects my lab is currently working on. So glioblastoma is the most common malignant brain cancer in adults. As you can see on the Central Brain Tumor Registry United States website, it always comprises a little more than 50% of adult malignant gliomas, adult defined as age 19 and up. And unfortunately, since the advent of the STOOP regimen, which is concurrent chemoradiation therapy, specifically using temozolomide for newly diagnosed disease, that was released in 2005. And we really have yet to still move the marker substantially for these patients in terms of their very abysmal survival, which still has a median of only about a year with treatment. Methylation the methylation status of something called MGMT, which is methylguanine methyltransferase, is very important when you're looking at glioblastoma research. So MGMT, if it's methylated, you kind of have an inactive enzyme. And so the DNA cannot undergo the repair that Temodar causes. So it actually, we found with the correlative study from the STOOP study, Monica Heggie and colleagues published that if you were methylated MGMT, no matter what we did to you, you actually did better prognostically. And that remains true, especially in the era of Temodar. Options at progression are very, very few. Patients' likelihood to be free from having another episode of tumor growth or progression once you first diagnose that is only about 85% or only 15% of them are not going to have progressed again at the next 6 months. So it's a very low threshold, again, low bar to try to beat. Moving on to connectivity mapping. So connectivity mapping is a bioinformatics tool that basically looks at your molecular profile of your specific cancer of interest and then compares that with a database of all of the molecular profiling signatures, what is downregulated and what is upregulated in FDA-approved drugs. And so it looks a bit like this. So you take your biological state of signature of interest here, and then in this case, of course, glioblastoma, and you run it through these reference database profiles. And what you find is you're going to have certain things that are very highly positively correlated and then negatively correlated as well. And in our studies, CMAP or connectivity mapping identified these top candidates looking at 4 different data sets. We saw in this very complex Venn diagram, there were 12 different drugs that actually found a good match for the glioblastoma signature specifically. Interestingly, one of those that really stood out was an HDAC inhibitor. In fact, there are a lot of HDAC inhibitors that stood out. In fact, if you look at this, of the drugs that were found, okay, 60 of them were HDAC inhibitors. So there's a long history of HDAC inhibition and study in glioblastoma. And unfortunately, as exciting as they have all looked in clinic or in lab, they've never really panned out in clinical trials, and not quite sure why. And so as you can see, this was the drug of interest. This was PCI, and it's now really known as abexinostat, made by Zynomic Pharmaceuticals. And here you can also see there are other, some well-known, there's trichostatin A, panabinostat, and then varinostat and belinostat. So a lot of HDAC inhibitors. So the question next that we asked was in our lab is, knowing clinically how that's disappointed, what's different about this one? Why is it showing up? Well, fast forwarding in the interest of time, we found the specificity specifically matches the HDAC profile in glioblastoma when you look at this new drug. So it HDACs 1 and 2 are upregulated in glioblastoma, and HDAC 11 is not. So the last thing you want to do is suppress one that's not necessarily upregulated, because what we found out is that actually helps to suppress tumor. So further suppressing that, which is what varinostat and belinostat and trichostatin did, you actually don't want. It also induces cell death agnostic of the MGMT status, P53, and EGFRB3 status as well. It decreased DNA repair machinery proteins as listed, and it was synergistic with temozolomide in glioblastoma cells. Finally, as shown in our orthographed models, it did improve overall survival substantially, with the purple line here being the PCI with temozolomide group. We did, in this grant, also look at varinostat with temozolomide because that had been the most commonly used one. I think it had 3 or 4 other clinical trials in glioblastoma that had failed. Then we looked at the animal models. We generated a genetically engineered mouse model that after about 4 months will actually get glioblastoma in its mouse brain. And this is with deletion of P10, deletion of P16, and the addition of oncogene EGFR variant 3, which is about 30% of glioblastoma have. And so looking at this mouse model, we're able to better recapitulate the true tumor environment of how this drug would work. And what we found was giving just the abexinostat alone prior to treatment and after 4 weeks of PCI, i.e. abexinostat, we actually saw about a 26% volumetric reduction objectively in this tumor. And that was with PCI alone, which also proved blood-brain barrier penetration for us. So with that, we have a clinical trial that's currently accruing. We're in dose level 2 of this phase 1 trial, and we use metronomically dosed temozolomide along with abexinostat in anybody with a recurrent, so grade 3 or grade 4 glioma. Hoping to close this up at the end of the year here. Moving on, second drug that we found was brompheniramine, and this particularly excited me. Those of you who treat gliomas might know these are one of the rare cancers wherein there's no relation to any risk factors, any lifestyle risk factors, whether you smoke or you drink or you exercise or never, really doesn't have any bearing. Only 1 to 2% of people have an increased risk because of a hereditary genetic cancer syndrome. But very interestingly, if you have allergies, atopy, or asthma, you have a decreased relative risk of getting this in your lifetime. So I've never been so glad to have to take Claritin every day in my life as when I found that out. And so this was very interesting because brompheniramine is an old, like 1940s old antihistamine. And so that was really interesting. So looking into that, the TCGA data set has a lot of patients with glioblastoma who were taking antihistamines. And interestingly, it's not a huge difference, but it is statistically significant in large groups. The people on antihistamines also seem to do better. So the case kind of grew. Then we found that HRH1 is very highly expressed on glioblastoma compared to normal astrocytes. Specifically, it's HRH1. So as opposed to HRH2, 3, and 4, it was really HRH1 that appeared to be the most consistently correlated. And this was true in human astrocytes. It was true in the U87 and 251 and 118 cell lines. And it was also true in the mice in our syngeneic mouse model and in our driven model as well. Here's a picture of HRH1 staining. And again, you can see that the relative HRH1 expression control versus tumor is much higher. So moving on from that, we said, what about combining this with treatment? And so let's try this with looking at specifically the U118 cells. So U118 cells are representative of the MGMT unmethylated. So those are the worst actors, relatively speaking, the ones that are even less sensitive to temozolomide naturally. And you can see in the U118 cell lines, when we gave brompheniramine, we actually saw that MGMT decreased. And further, we did see increased DNA damage as well in the form of phospho-gamma-2HX, which shows us about strand breakage. That's also shown here in the picture lower right. You can see the temozolomide plus brompheniramine group. And where you have the bright pink, that's more DNA damage. So we did see none at all with control, barely any at all with brompheniramine alone, a little more temozolomide, but the combination showed significantly more. Then moving to the animal models, temozolomide plus brompheniramine decreased tumor burden and significantly improved survival in vitro. These are our mouse models that you can see here. Control after 30 days, significant growth. Temozolomide alone, not a whole lot better than control. Brompheniramine, perhaps a little bit, kind of hard to say. And then temozolomide with brompheniramine definitely appeared more successful. And when you see this objectively in the photon flux graph here, you can see that there was a significantly better suppression of tumor at 30 days in the temozolomide plus brompheniramine group there in the green. And here we have it on a Kaplan-Meier curve compared with the temozolomide here in purple, brompheniramine alone in red, and then the green with the combination. So we're very excited about that, and we're actually resubmitting an NIH grant to look at that a little more deeply, and then also to have a correlative clinical trial with that. So finally, I'll move to fedratinib. Fedratinib was yet another one that showed out. Now, fedratinib is used by my colleagues who treat leukemia, so that was certainly interesting. And we know that the JAK-STAT pathway role is pretty important in glioblastoma and has been established previously. STAT3 expression and phospho-STAT3 signaling is highly active in human glioblastoma. And this really seems to promote more of aggressive phenotype and therapy resistance as well. This is a bit of a busy slide. I'm going to gloss over this a bit, but basically it promotes glioblastoma tumorigenesis by many different pathways, including local immunosuppression, survival, proliferation, migration, invasion, neoangiogenesis, makes it more radioresistant, which we'll get to at the end here, and chemoresistant as well. So in the cell line studies, again, starting with our cell lines, the combination of treatment of temozolomide with fedratinib decreased proliferation in the U251 and then also the U118 glioblastoma cells and increased apoptosis. Not all of the figures are shown. The combination decreased STAT3 activation as well. It decreased spheroid-forming invasion capacity of cell lines, and that's shown here. You can see the combination treatment versus the control versus temozolomide alone as well. And then finally, we also saw that it decreased proliferation and stemness markers as well in mouse syngeneic cell lines. And finally, also shown on the lower right is that it decreased the DNA repair enzymes. And again, there's that MGMT that we worry so much about in glioblastoma. Finally, we also found that radiation, we know radiation increases STAT3 activation. And so when we look at different cell lines, and these are from some of our syngeneic cell lines, two different just examples here, particularly one here, you can see that phospho-STAT3 increases with increasing amount and exposure to radiation therapy. And so Dr. Zhang is now working with us in lab. And what we're trying to figure out is a good way to harness, upfront glioblastoma we treat with concurrent chemo and radiation therapy, and considering developing a clinical trial to use this upfront for newly diagnosed disease. So what's next? Our lab is looking at the effect of fedratinib on patient-derived cultures. Why fedratinib? How does this compare to other FDA-approved JAK2 inhibitors? Is there one that maybe has a better toxicity profile, for example, that might give us the same responses? And then we're obviously going to evaluate the combination with radiation therapy as well. So that's kind of a whirlwind tour of everything that we're doing really in the lab. Thank you all for your attention, and I'd like to thank our funders. Stop the show for the next person.
Wow, that's impressive. I wish all three drug works. So we have three more options. All right, so we have the next speaker, Dr. Michelle Isenberg. She's, again, I guess, someone I can never get off my back. So she's currently the professor at neurosurgery, has been mainly focused on primary brain tumor surgery and a lot of neuroconnectivity research using brain MRI, functional MRI. So she had a talk quite a while ago for us. So really, hopefully see something more exciting this time.
Wow,
Oh, boy. Good afternoon. High expectations now. It's a pleasure to be here. I do hope you find it exciting and interesting. I do. I'm going to talk to you today about some of the work that I'm doing. Today, we're looking at accurate prediction of individual brain function maps from structural imaging utilizing deep learning. This is my disclosure. So what we're going to go over today is first, we're going to review brain mapping. What is it and why do we use it? I'll show you some data that evaluates the accuracy of clinically available methods. And when I say clinically available, that means things that a clinician has readily available to them in standard clinical care. We will assess an alternate method of function localization utilizing deep learning and discuss briefly clinical applications of that. So first, what is brain mapping? So brain mapping can be invasive or non-invasive. So first, we're going to look at non-invasive mapping. We want to look at functional areas in the brain, and this is used as a neurosurgical planning tool to help us identify the relationship of the tumor to these eloquent areas so that we can protect them. So important areas we may want to know about would involve language function, motor function, sensory function, or possibly vision. And ultimately, brain mapping helps us achieve a maximal safe resection while attempting to prevent the negative effects on quality of life, morbidity, and survival times. So let's look at some of the standard, I'm calling them clinical techniques. And some of you may be familiar with these and others may not, but we will go over these. The first is functional MRI. And a functional MRI is when a patient lays down in the scanner and data is acquired. A functional MRI can localize where a particular action is taking place. And this can be task-based, where the patient may be asked to tap their index finger or their foot, for example. And what this study does is it measures changes in oxygenated blood flow. And this happens when a particular brain region is activated, more oxygenated blood is sent to that region. Now, another version of this is resting state, where the patient lays there and, like it's called, just rests. They let their mind wander and aren't focused on doing any particular task. And what we see here on the right is an axial image showing the functional MRI activations mapped onto the anatomic image. This is the area of tumor here, and the blobs, the colored blobs, correspond to a particular activity. For example, purple is left motor foot, and as an aside, I would not expect it to be showing up where it's showing up. And blue is left index finger tapping, which does show up where I would expect it to. Another available method for brain mapping, but I'm not going to say it's readily available, is magnetoencephalography or MEG. The reason I say it's not readily available is because many centers don't have this. We fortunately do, so it has been a big part of my brain mapping program. What MEG does is it records the small magnetic fields that are produced by electrical currents in the brain. And like functional MRI, this can be done in a task-based or resting state. And magnetic field changes can be analyzed to estimate the source of these changes. What we see here is another axial image, a different tumor patient, and the tumor is located here. And again, we have colored, these are dipoles. This is a processing, which is the clinical standard for MEG, is dipole analysis. There are various ways of analyzing the MEG data, but again, this is dipole analysis. So you can see that the aqua is sensory foot. This is the area of tumor here. I wanted to know its relation to sensory and motor areas. This is where I would expect that function to be located. The sort of hard to see motor foot in magenta is right here, again, where I would expect it to be. And then out here is sensory hand and motor hand. This is what the MEG machine looks like. It's sort of scary, humongous, gigantic hairdryer type of thing. But it otherwise looks fairly comfortable. They get to sit up instead of laying down. So that's maybe a benefit. So another way that sort of indirectly localizes function is through tractography. So diffusion imaging is processed to give us tractograms. And diffusion imaging provides information as to the integrity as well as the directionality of groups of neurons in the brain. So tractography can be used to visualize important tracts of interest. For example, the corticospinal tract, which is for motor, thalamocortical for sensory, the arcuate fasciculus, the major one involved with language, and the optic radiations for vision. So the diffusion images are processed. This is a DTI map showing the colors, which again indicate direction of the fibers. When the data is processed, we get a tractogram, and you can see here this person has a tumor, and I wanted to know its relation not only to where motor may be cortically located, but where is that important fiber tract, because I want to try and protect it during surgery. And this study indicated to me that the corticospinal tract was most likely posterior to the tumor. Of course, I would verify these findings in the operating room. And this kind of data that I've been going over with the fMRI, MEG, and the tractography can be brought into the operating room with us. So speaking of the operating room, takes us to invasive mapping. And this is where data is acquired directly from the brain. And therefore, it is considered the gold standard in brain mapping, in localizing function. This can be performed with the patient awake or asleep. And there is stimulation that occurs, usually at the cortex, but we can also do subcortical mapping, which can help localize or tell the proximity of the tracts to where we're working and be helpful in that way. A sleep mapping can be done for sensory or motor, but language mapping can only be done awake for obvious reasons because there's no other way to test how something is working. So this is a picture of one of my patients who had awake mapping. This area here with stimulation elicited activity for motor function in the face and throat. This area here, when stimulated, resulted in speech arrest, and there was interruption of function here with some speech hesitation or slowness. So I want to take a moment, and before we go into talking about how accurate are these methods, and just have us think for a moment about reliability and accuracy in science. So when we are creating experiments or tests, we ultimately want to have something valid, right? So over here on the left, using these bullet examples, this mouse arrow keeps disappearing. This one here on the left is showing something that has poor accuracy and poor reliability, not something we want. In the center here, we have something that is giving us reliable results, but if we don't know that it's not accurate, then this is going to be flawed. Over here on the right is ideal. We have something that's reliable and also accurate, meaning it will give us this information accurately over multiple times that we test it. So I wanted to know how accurate is fMRI and MEG in localizing function. Anecdotally, I would get results that were not what I would expect. So it would show me function in areas, as I showed you earlier, that seemed almost spurious, or I would localize something in the non-invasive mapping, and then in the operating room, I would find the opposite, maybe no function. But it's good I tested it because then I knew that that area was safe to take. So in a cohort of patients where I had data that included fMRI, MEG, and intraoperative mapping, we looked at the accuracy of fMRI and MEG using the intraoperative data as the ground truth. And what we found was that MEG analysis was more specific. This is looking just at motor function, was more specific than fMRI, but it was less sensitive. And for language function, we saw something similar where MEG was more specific, but less sensitive. So having some confirmation of what I was seeing, I had a question of, is there a better way that we can elicit this task location from imaging in a way that is going to be clinically useful? And we thought about how task fMRI can be predicted from resting state fMRI and thought about another imaging sequence that could be helpful, such as diffusion MRI. Some of the benefits of diffusion MRI is that diffusion images are routinely acquired, not only with standard brain tumor protocol scans for surveillance, but also as part of our preoperative image set that we acquire for navigation. It does not require any task performance by the patient. So if somebody has deficits, for example, and maybe they have a hemiparesis on the side that you want them to do something, you could still get a result that's useful, that you could potentially trust. Also, diffusion has high test-retest reliability. So what we did is trained an AI model, a 3D convolutional neural network, to predict functional brain activations from structural brain images in healthy subjects. I don't know how many of you may be familiar with the Human Connectome project, but we utilize data from that. They have many patients. These subjects had functional MRI data as well as diffusion data. So we trained a 3D UNet convolutional neural network for our model, and then compared our predicted activation maps with the actual activation maps. And 47 unique task activation volumes from 7 different task domains were used for model training and testing. Now, we don't test all of the different functions that they did, but it was nice that we had many of them to look at. And again, these are healthy subjects. So we evaluated the ability of the predictions to differentiate between individuals by using a correlation matrix. The predicted task activation maps for a given subject were compared to their own functional MRI maps, as well as to all of the other subjects' maps, and a correlation matrix was constructed. So what we found was that the convolutional neural network was able to use structural imaging to predict activation patterns of individual subjects that matched the group average, but also could accurately map activations that deviated from the group average. And so what you see here in this column is the group. Subject A is someone whose actual activation maps fit along with the group average. Our model predicted accurately where the activation maps should be. Subject B is someone that has a more deviant, not deviant in a terrible way, but activation maps that deviated from the norm, let's say. And you can see here, we've circled areas of interest that our model was able to predict these activation maps as well. So we were pleased with that, but we wanted to know if these predictions are going to be reliable when tested on repeated scans. You know, is this a one-trick pony, or is this something that is going to be reliable? So what we did is evaluated subjects that were scanned 4 months apart. And looking at an interclass correlation coefficient, we saw that the activation predictions with our model had excellent reliability for all of the task domains. The actual fMRI data performed worse with only good to moderate reliability. So this was good and interesting. So our IM model produced highly reliable predictions of task activation while also detecting individual level variations in healthy subjects. But I specialize in brain tumors, so I would like to know if this can be applied to patients who have brain tumors where your anatomy is distorted. So to see if eloquent function location could be accurately predicted from diffusion imaging in brain tumor patients, we performed another experiment. We trained a similar model to predict the location of motor cortex. So we didn't look at a bunch of different tasks, we just focused on motor. We looked at the location of motor cortex from the diffusion imaging in healthy subjects. We trained the model from that, like before. This model was then applied to data from brain tumor patients from where I had data. The intraoperative mapping was again used as the ground truth. Results were then compared to task fMRI as well as other mapping modalities. So this is an example of what we saw. This patient has a large left frontal tumor in the posterior frontal area. Again, we're only looking at motor, but I did look at language in this patient as well. But you see the tumor here. These small yellow spheres are the intraoperative mapped points for motor function that I found. And then the red here is showing you the DTI CNN motor predictions. And indeed, it's where it should be based upon the ground truth of the intraoperative mapping. So what this is showing is that our model outperformed all of the other mapping methods, including the task fMRI and the task MEG for motor mapping in this cohort of brain tumor patients. So this was very exciting for us. So again, the results show that predicting task activation from diffusion produces more reliable and accurate mapping results than standard task-based fMRI. So in conclusion, this preliminary work shows that a deep learning model can use structural features to predict brain functional activation locations. In brain where structure is disturbed by tumor, the model accurately predicted the location of motor function. Now, we need to do more work to expand this to a larger cohort of brain tumor patients, but also look at other tasks as well. And this requires a lot of data, so this may take a little time for us to get, especially if we are using intraoperative mapping as the ground truth, because not every brain tumor patient who has surgery and may have non-invasive mapping is going to get intraoperative mapping. When this is further developed, this methodology could potentially be useful when eloquent area information would be useful. I've been talking about it for neurosurgical preoperative planning, but also for SRS planning, I think would be very useful. And also in situations where longitudinal assessment of task location would be interesting to look at over time, neurodegenerative disorders, or in my case, I'm interested in how the brain reorganizes and how different connections are made over time from the tumor growing or from treatments. So I'd like to acknowledge Mr. Ellis, who is in my lab, who next year will be Dr. Ellis. He is the one that makes all of this magic happen because I certainly don't know how to do this programming. And that's it. Thank you for your attention. I hope I didn't let you down after Kevin's expectation.
Well, we have 4 talks today, which is a little more than usual, because I did volunteer myself for the talk in addition to be a moderator in case we have issues. So I do have to say my talk compared to today's keynote speaker is probably going to be quite preliminary, but I'm happy to share some of our studies that we performed lately in our group. And first, I'd like to thank also Dr. Isberg. That's quite interesting. That's definitely not disappointing me at all. And also thank you for setting a very nice foundation for using the AI technique, deep learning technology. So as you all know, I'm actually more a lab person. I'm more interested in testing drug in the animals, in the cells, but artificial intelligence, this is such a calling concept. It's hard to resist the temptation these days to do anything. So we did some preliminary study just to prove the concept. So today I'd like to talk about this study we did using artificial intelligence to try to assist the early diagnosis of very small brain tumors or brain metastasis in particular. The idea is to try to see whether AI can exceed the sensitivity threshold of human eyes for diagnosing those brain metastases. And here's my disclosures. And for brain metastasis, I think everyone knows this is the most common actually brain cancer in adulthood, way more prevalent than the primary brain cancer. So we do see more patients in the clinic than treating GBM. Every year in the United States, we're talking about 100 to 200,000 cases probably diagnosed per year. And the early detection of the brain metastasis is definitely vitally important because it renders patient more likely to be eligible for the so-called stereotactic radiosurgery instead of being treated by the older technique such as whole brain radiation, which cause much higher level of neurocognitive deficit and less chance of having durable tumor control. And also its potentially can avoid the need of multimodality treatments such as surgery plus radiation, which also will be more toxic. But how do we achieve this higher sensitivity of human eyes to detect small brain mets? I think the easy question will be, well, let's make the imaging quality better. So we could use a 3 Tesla MRI scanner instead of 1.5 to probably detect smaller ones. But I would say the hardware improvement, there's always a ceiling of limitations. I think ultimately, in my opinion, the human eye sensitivity to the small object probably will still be the ultimate limitations. So the idea is whether we can use AI to improve our shortcomings. In this field, particularly for brain metastasis, we saw a lot of studies, but most of the study are focused on either differentiating the brain mets progression versus the radiation-induced radionecrosis, or trying to give some indication for primary sites for newly diagnosed brain mets, or sometimes trying to differentiate brain mets, solitary brain mets from like a primary brain cancer. But we are more interested in seeing whether the AI technique can help detecting early brain mets. So the hypothesis we have here is there is possibly a high risk of missing diagnosis of small brain metastasis, even by a trained, experienced neuroradiologist based on brain MRI alone. And if that's true, the AI technology could potentially detect imaging features way early beyond the human eye's limitations. And this is a hypothesis we generated based on some of the clinical experience, because sometimes we do see when the patient referred to us for SRS, sometimes we could pick up small ones that was not called by the radiologist. So our short-term goal is to really estimate how high the risk is in our practice of missing the early detection of the small brain mets, and also to analyze some characteristics to see whether we can identify something that we can improve in clinic right away. But in long term, we would like to develop some AI plus minus radiomic technology to facilitate the identification of the very early brain mets for future improvement to reduce the human error. So our methodology is quite straightforward and quite simple. We started to connect patient data for those patients referred to us with brain metastasis diagnosed since 2015 up to 2020. The reason we had a cutoff of 2015 is that's a year we started to universally apply the radiosurgery protocol for brain mets patient for high resolution sync-cut T1 post-contrast sequence. So we connected all those patients, and our focus is mostly for those very small brain metastases. And as you can see on the right side, this is a large brain mets with very significant vasogenic edema associated. And I definitely hope our neuroradiologists won't miss this one, which that never happened. But for these very small ones, sometimes they are also located very close to other structures with contrast enhancement, such as the blood pools and the box, which has rich vasculature that could potentially distract the human eyes, even it's visible. And in this tumor, luckily it has quite significant like flare hyperintensity, which can help the detection of this small lesion by looking at other sequences. But for many of the small ones, you're not always lucky to see a significant flare signal. And also some of the flare signal T2 hyperintensity are mixed within those neurodegenerative change when we see in the aging patient population. So our study are pretty much focused on the high resolution T1 post-contrast sequence to look at the brain mets. And the inclusion criteria for this database is we included all patients in that period for a newly diagnosed brain metastasis. No primary brain tumor is included. No patient should have leptomeningeal disease. For each newly diagnosed brain mets, the patient should have a previous MRI brain scan about 1 to 6 months before the so-called diagnostic time. And no prior history of whole brain radiation or prophylactic cranial irradiation is allowed due to the concern of the whole brain radiation potentially changing the imaging feature of those scans. So once we connect all the data of the patient information, we grouped all the MRI scans into our Velocity, the imaging processing software, and have our students, including even high school students, medical students, to help to review those imaging using their naive eyes. So we're not purposely not trying to get a neuroradiologist to do this. So this is one example of a patient that has a newly diagnosed brain metastasis. As you can see, this contrast enhancing lesion located in the crosshair area. And by looking back to the fused MRI scan performed 3 months earlier to localize to the same area with focused review of this area. For example, in this case, we can see a very tiny dots, which is contrast enhancing. That was actually not called 3 months earlier. So for this case, we classified it as a patient with a missed diagnosis. And also the criteria for all these patients that we consider to have a misdiagnosis are they should have a visibility of a contrast enhancing solid mass or lesion using the highest magnification of the imaging in our software. And we should be able to detect at least in 2 axes contrast enhancing lesion, if not all 3 of the axial, sagittal, and coronal view. And if we have doubts still, then we'll look at the contralateral side of the brain to make sure there is a difference. So by going over all those imaging and the cases, we actually connected 55 patients with a total of 126 new brain metastases in the range of year 2015 to 2020. And altogether, we have 411 MRI scans reviewed. It's quite impressive for us to ended up seeing that for all those lesions that claim to be newly diagnosed, when we look at that particular area, we actually see about 44% of the lesion that actually can have a contrast enhancing lesion that retrospectively visible. Because the average number of brain mets per patient is 2.3, that kind of makes the percentage of patient affected by missed diagnosis even higher, being about 56%. But fortunately, all the missed brain mets are very small. We measured the size, the maximum diameter of the size of those missed metastases. The average size is only 3.0 millimeter, ranging from 1.2 to 7.7. And as you can see in the size distribution, with the size increase, the number of missed metastases drop exponentially. We also did some preliminary study, look at all kinds of clinical factors to see whether we can find something that predict higher chance of missing the diagnosis. The gender, primary cancer type, location doesn't seem to have any significance of predicting the chance of missing. And consistent to the literature in our database, we have most of the brain mets started from primary lung cancer. And also numerically, we find that the brain mets to the cerebellum seems to have the highest chance of being missed from the early scans. And the one phenomena we caught our eyes that seems to be interesting is that a significant portion of those missed metastases seems to be located close to some contrast enhancing structures. For example, dura, blood vessels, sinuses. And this is one of the examples that's showing you that this patient had a so-called newly diagnosed brain metastasis located in the left frontal lobe, very much to the surface, close to the meninges and dura. And when looking back 1 month earlier, we saw that contrast lesion look actually visible. And this lesion actually sits just in a mirror location to the contralateral side of a vasculature. And I think that possibly contributed to the distraction of the viewers and had been missed. And this is another case that we saw a patient had a brain mets from renal cell carcinoma that was treated with SRS. And 3 months later, we had another MRI to follow up. And the treated lesion did show very good response, but there is a new lesion popped out, again, quote unquote, in posterior portion, posteriorly to this treated one. And then going back 3 months earlier, yes, we still again can see a lesion that's already there, but very small, and it's located among all the vasculatures. So again, locating close to be treated large mets among all the blood vessels probably again distracted the reviewer of our radiologist. So I think from these studies, we do confirm our concern that there is quite a high chance to miss very small brain mets. And I'm not saying our radiologists are bad. That's, to be fair, that's not true. It's only because many of the lesions, we need to blow up the image so big to be able to identify it. And also, this is also why we say hindsight is always 20/20, because if we know where the high-risk area is, and if we just focus on that area, even an untrained high schooler or a medical student can easily outperform a very well-trained neuroradiologist. So the issue is, how do we find that high-risk area? So we carried out a project with the help with Dr. Shuo Wang, one of our medical physicist team, to develop this AI assisted early detection algorithm. So starting by having the student contour out the brain mets in the MRI and identify the center of that new mets and match that center to the early MRI a few months before, expanding that single point by 1 centimeter to generate this so-called prior to growth volume. This is generated in the pre-MRI that was not supposed to show any tumor. And we copied this lesion, this volume to the contralateral side to generate this contralateral negative volume as controls. And this right side image is just showing you that if the location is very close to the midline, we need to chop off the expansion portion to the other side of the brain so that these two volumes do not overlap. So by doing that, we also proceeded with this deep learning model using the 3D convoluted neural network algorithm as well, which similar to what Dr. Eisenberg was discussing earlier. So I probably don't need to mention again, but we input the 2 different sets of the imaging into the algorithm based on this multi-layered approach, and eventually filters through all the parameters into this flattened extracted feature blocks, and eventually after dropout to increase the generalizability, we can have a binary output based on the 2 different sets, basically like 1 for high risk area of the prior to tumor growth imaging and 0 for the contralateral negative control. And with this data set and the algorithm, we divide our data to the training set, validation set, and the prospective testing sets. And based on that 121 lesion that actually has analyzable data, we achieved accuracy of prediction of the high-risk area to be accuracy of predicting either negative or positive area to be about 69.2%. And this relatively low number is mostly contributed by the very poor accuracy of predicting for the contralateral negative side, which is only 38.5%. So to improve the accuracy, we did something called data augmentation by simply rotating the area of interest box every 18 degrees to generate 9 times more data sets for repeating the same analysis. And using much larger data sets, we actually achieved 100% accuracy in both predicting to the high-risk area and the contralateral negative side. So this is just a slide showing you the so-called gradient weighted class activation mapping of the grad cam on the tumor side. So the idea is based on this heat mapping area, it actually tells us how to understand what is the location that AI is paying attention to or focusing to, or in other words, what's the most characteristic area that the AI did a classification. So probably to be a little easier to understand, I put this golden retriever picture here. If you want to use the AI to try to tell this is a golden retriever, and then the end result of the heat map is located around the ear and the eye area that indicate this is the area that's most characterizable for telling this is the golden retriever, at least to the eyes of this AI algorithm. But still, we don't know exactly what that area means. It just gives us an idea what's important for the algorithm. So in conclusion, we did detect pretty high rate of missing human error, missing the early diagnosis of brain mets. And based on our data, it seems that if the tumor is smaller than 3 millimeter, the reliability become pretty poor, even in the trained eyes. And also we find significant portion of patients with this missed metastasis was probably located in some distracting areas such as the contrast enhancing structures. And in our hands, AI does seems to carry a potential to possibly function as a powerful tool to assist the human eyes to detect the brain mets early. The limitation is obvious because all the speakers today talk about the sample size, the validation, the necessity for external data sets validation. And we obviously would like to continue to do that. And we're collaborating with external collaborators to try to get different source of imaging to confirm this. And also one issue for AI, as I mentioned earlier, is it seems like the AI is providing us a kind of a black box of the algorithm, although it can predict what we want to, but we just don't know how it predicted. And in our model, I think there are two scenarios that human beings can miss the diagnosing of the early brain mets. The first scenario is we could have the tumor developing with some kind of high-risk feature in that area, but that feature is under the threshold of being detected by human eyes. The second scenario is the tumor can be visible, but is located in an area with strong distraction. And we don't know what the true distracting factor is to the eyes. And using AI approach, I don't think we can truly tell these two parts up differently. So we're planning to study on the same data sets, looking at the radiomic features and hopefully try to select the stable and predictive models to further understand what's the reason to miss the diagnosis. And finally, I'd like to thank our team, particularly Dr. Justin Burr, our chief resident, who led the team, including the high school student, the medical students, and Dr. Curtis Johnson for contouring, reviewing all the imaging, and Dr. Shuo Wang's team for sure for developing the AR studies. All right, so now I'm putting my moderator hats back on. So we have 20, we should have, yeah, let's have all the speakers come back here. I think Dr. Shankar need to go back to have another meeting, so probably we'll forward the question to her later. So I guess we'll go by the online question first. I'll probably read it out first. So with the application of AI, we now know a lot more in the molecular stratification of brain tumor. And are we ready to use the complicated information for treatment? I think that's a question for Dr. Chen.
This is a great Great question. Maybe we can even answer this question in the group. Yeah, I think so. At least now in our daily practice, we are definitely using those complicated informations. For me as a neuropathologist, I definitely use those information to guide my diagnosis. And then whatever diagnosis I gave, and then my EGFR amplification is actually one of the 3 molecular features of GBM. So I would have to, upgrade my diagnosis. It's not a grade 2, let's see, astrocytoma with IDH wild type. I have to call it glioblastoma IDH wild type, CNSW grade 4. And then we discuss this case and tumor board, and then we will see, by imaging, by morphology, it's low grade, but now I have molecular information that I have to call it grade 4, I'm sorry. And then our oncologist will treat this patient as a grade 4 GBM. So I think, definitely, now here at UMC, we are, using those molecular features to, guide our diagnosis and treatment.
Yeah, I think that's becoming quite a common theme these
Yeah,
days.
yeah.
But one drawback is kind of delaying the starting of the adjuvant treatment quite significantly.
Yeah,
We're waiting
yeah.
for all those markers to come
Yes,
back.
yeah.
So hopefully in the future, maybe even AI or the imaging
I think,
diagnostic modality.
I think somebody is actually working on that. Just try to get some morphological features to predict the molecular, let's see, the methylation classes. So I believe, some studies have been published on that topic.
I guess in the future, maybe even like diagnosing the tumor even before the surgery, then you could guide surgical approach too.
Well, I was gonna say that even from a surgical standpoint, if I see someone that may have had a biopsy on the outside and I know what it is, oligo versus astrocytic, although that's a little more simplistic, but my operative thought process may be different. Conversely, if somebody has a bad actor, that may influence the decision to take to the OR, where other treatment, which is equally important, would be delayed, making things worse for them. Especially if maybe they have multicentric disease, where I may say no, or if somebody has multicentric disease, but something more favorable, then maybe I take out both areas. So I think it applies for all of us.
Yeah, so let's move on to the next one. Thank you again, Dr. Chen. So for patient whose tumor is downgraded from GBM to another entity based on methylation profiling, is there sufficient data to conclude that patient with downgraded tumor live longer than patient with GBM? Or is the number of cases available too small to draw any firm conclusion?
Good question. Yeah, so I think the original 2018 study did not look at particularly the prognosis of those patients. But based on my own practice, based on the cases I see, I think I would imagine that those cases, those CBM cases that have been downgraded to, let's say, PXA would have long survival. I'll give you one example that I saw. So we had a patient who was initially diagnosed at the outside hospital with GBM 10 years ago. and then came to our hospital to see Dr. Bianca, and then I reviewed slides. So I looked at the slides without any information. I even didn't know the patient age, sex, and anything. I look at the slides, I'm like, it looks, I need to think about PXA. And then, so I started all those molecular workup. That tumor was positive for BRAF. And then I sent for methylation. It indeed matched to PXA. And then, so retrospectively, we know that, so that patient, did survived much longer than we would expect for GBM. So, and then I'll give you another example. So for the patient I mentioned, the HPAP, the another entity, I did see a few other cases at the
And then with the new pathology is the IDH mutant. So now we don't call it GBM anymore.
Correct.
But then the question is, does that really change the practice? Because it can be downgraded, but we know they are doing better because we treat it more aggressively. But are we downgrading the tumor but downgrading the therapy? So far, I don't think that's the case. So
they've already been treated.
well, that's true too. But even prospectively speaking, we're probably still gonna be aggressively treating IDH mutant astrocytoma grade 4, similar to GBM, because we know if we do that, they do better.
The data, which can include tractography and or fMRI. I think the likelihood of MEG in the RADONC planning is unlikely, but certainly possible as well with the same software. It might require obtaining another study because I've done a lot of work co-registering imaging pre-op and post-operative at multiple time points. And it has some challenges. You saw in my talk, one of the renderings showing the DTI CNN predictions in the brain tumor patient. That's an example of a rendering. So if something could be developed like diffusion imaging and task localization, it would be much easier, cost efficient, time efficient to do that. But generally the answer is yes. To actually have it as part of the treatment plan actively, that data just would need to be imported in and essentially be an overlay in the image set that you could turn on and off.
Yeah, so I think that's probably gonna be the more standard future practice. But if you can demonstrate the diffusion imaging is that good, yes, then we definitely will commonly routinely use it. All right. So Dr. Eisenberg, would you advocate for functional MRI for all of your gamma?
Well, I'm going to assume gamma knife is just referring to stereotactic radiosurgery. I wouldn't advocate it on every single one. I would advocate it on anything that is pericentral, meaning, around the frontal parietal area or in the frontal operculum. Even possibly on the non-dominant side, language can sometimes be co-dominant, or in what we call the receptive language area or Wernicke's. So posterior, superior temporal, lateral, inferior parietal area, and also vision. And that could, so every time a functional MRI is ordered, the DTI or the tractography is also being processed. So you get the tracks and the activation maps.
So that's actually bring a question offline here. Like your gold standard is intraop,
Mhm.
but how much deviation do you call it a true deviation?
Yeah, that's a great question. A lot of the literature uses a centimeter. So within a centimeter would be considered accurate. And like centimeter, well, that's
See, that's a problem.
that's the next gyrus, And so it's very tricky. And our paper talks about this and looks at that. And there's different ways of thresholding the data, the fMRI, for example. And so there's a lot of different factors, but
Yeah, but that's kind of bring an issue. It's just like if we're talking about gamma knife accuracy, if you have 1 centimeter. Sites, then the leptomeningeal disease probably will be more interesting to us. Because if I can predict in a future study, we can get leptomeningeal spread in the next 3 to 6 months, I might offer PCI or whole brain radiation instead of still doing localized focal radiation. But those again, the beauty of AI is does open up the infinite possibilities. I think Dr. Shankar has left for another meeting, so we'll probably save this question for her. We'll probably deliver this to her next time. The last one, since AI can identify lesions that human missed, do you think that AI will replace radiologists in the near future? I think I probably chip in first, you guys.
well, it's for you.
Oh, that's fine. But you guys should contribute because it's the same thing. Like if the AI can predict if this patient gonna die in two months, even given a brain surgery, we just still operate. But that's later. I guess I would probably quote what Dr. Fuller said. Because ultimately it's a sense of responsibility there. If the AI can claim medical responsibility, yes, then we don't need a radiologist. Anytime you need a billing, you need a consent, you need any patient interaction, I think radiologist's job is still there.
That doesn't make sense. He was talking last night about the type. There was the Astro one where I think it was for liver tumor tracking for like hepatocellular. It was very accurate, and then they said, okay, they gave another CT, but it was a brain CT. Said, where's the liver tumor? Go, oh, it's right here. So,
again, the tricky part is how accurate the AI algorithm is going to be in the future. Maybe it will replace a lot of work we do. Just like just like the example he said, think about the anesthesiologists, what they do these days. They sit there during the entire operation, look at the monitors. They don't pipe, they don't pump, they don't walk. But we still need anesthesiologists.
Wow.
Okay.
I think that for all of our flaws as humans, that we still have a lot of valuable assets in terms of interpretation and processing of information. I think AI is extremely helpful, but it is not absolute. There are times when I think the combination of the two would be helpful. Not every lesion seen on imaging is a MET or a tumor or a stroke or an infection. And so AI related methodologies can be helpful in identifying what a lesion may be, just like maybe spectroscopy, which isn't very good, but I'm just using that as an example. It's an adjunct, or perfusion. So I am afraid of the time where there are no surgeons, there are no people reading something and doing some interpretation. I think to get to a point where humans are even close to not being needed, the amount of training and information that needs to be processed to create a model to be able to do the things that our mind does would be quite vast. And it certainly may come to that, but that's just my 2 cents.
Not in the near future.
Yeah.
Also, I think one thing I want to add here is, I think AI is great in performing routine tasks. Let's say stuff we already are very mature, very highly standardized. We should dump that to the AI. I don't think I should sit there and contour the whole brain all by piece by piece myself. But the human brain is so unique that it does catch up something abnormal way better than anything else. Because I think, for example, that golden retriever picture, doesn't matter how many times you train how you train the AI. If you never train this AI to differentiate a golden retriever from a lab, when you put a lab picture there, AI is not going to differentiate that. But as human, even for a baby who never seen a lab before, I'm pretty sure he can tell the difference. So we do have to pay attention to the difference of human model and the AI world. And I think humans always have a role in this earth, on the earth.
I mean, we're not egocentric at all, but.
A couple of questions. I have one question for Dr. Eisenberg and one question for Kevin. So my question about this mapping is that when you have a discordance between fMRI and intraoperative mapping, so you're not going to trust you still trust intraoperative mapping, right? So if that's the case, why do you even do the preoperative as opposed to just, do everybody just do intraoperative mapping?
So the question for those who may not have heard it is basically, why even do any preoperative assessment if intraoperative mapping is the gold standard and you're going to go by that anyway? Not everyone actually needs to have intraoperative mapping. Even though I love doing it, there are some different risks that come along with it, at least for awake mapping. Language is not discretely organized like motor and sensory function is. Plus, there's reorganization that occurs, and there is data to support that the combination of the preoperative non-invasive mapping along with the intraoperative mapping does provide better outcomes than just doing the intraoperative alone. So generally speaking, I would go by my intraoperative findings, but even intraoperative mapping is not always clear cut. If somebody has a seizure, for example, I'm not going to continue the stimulation mapping. Then I rely on my clinical exam that I saw pre-op. What did the non-invasive mapping, show me what was the neuropsych testing, and then I have to make a decision. Am I going to touch this or am I going to leave it alone? And so not having that study beforehand, I think cheats me and the patient out of important information that will help make a better, safer decision.
I have a question for Kevin. So, the concept of early detection for metastatic disease has been debated for decades, right? So we don't actually know what that means because metastatic disease is metastatic disease. So the size really doesn't matter. That's why if you look at staging, the primary disease, if it's localized, you can have a T1, T2, T3, T4 based on the size. But once you have M1 disease, it really doesn't matter when you have a 2 millimeter one or 4 millimeter one. So I think the model, that's interesting that you have a model that you can detect early metastases, but the question is, whether that's relevant, right? You have a 1 millimeter metastasis. So I think it's very more important, actually have a model to detect the primary brain tumor because that size, that really matters, right?
Yeah, I think that's a good point. I think those are probably two different questions. One is, what's the value of doing early detection for brain mets that's already staged, patient at stage 4? You're probably not going to change the treatment modality much. But I guess one major reason we're trying to do this is for cost benefit issues. Because these days, if you follow the strict guideline, if patient has a brain mets, we're supposed to scan them every 3 months with the MRI. And imagine how big a financial burden it is to the patient, to the society. And we're talking about patients live longer and longer. They live 2 or 3 years these days easily for many primary sites. And we're talking about each MRI, if we're paying out of pocket, $7,000–8,000. And who's paying that? We're paying that. So if we can develop some algorithm, not only just early detect, but predict, because in this database, we are still lumped all the negative pre-diagnosis MRI with very tiny lesion together. So the next step is we're going to tease out the ones that's visible. Just look at the invisible one and see whether the algorithms still work to actually predict whether there will for sure be a new lesion in like up to 6 months from now. So if the algorithm become very accurate, then we can skip the next MRI. So potentially we can cut down by 50% of the need of MRI if we do that. And think about that. They're talking about 15% cut for radiation oncology.
Yeah, I understand, but my question really is that probably is not going to change the management because if you see 2 millimeter lesion, you still have the option of observing that patient, right?
Yeah, that I agree. But I guess to us, this geographic areas probably make more sense because you're not always expecting patient to follow up with you reliably. So let's say if they missed the appointment for one time, that elongate to 6 months. And if we missed, let's say a 2 millimeter lesion, next time is probably like 5 or 6 millimeter. But if you miss that one, next time is probably 1 or 2 centimeter. And then that's what makes the treatment possibly quite different. And also the patient, once the patient starts to become symptomatic, then you add another layer of the medical cost. Not only symptomatic management, but hospitalization, all those things that could have been prevented. So I think that's the first concept of the question. But your second question, I almost forgot. In terms of the primary brain tumor.
Yeah, so that is the point that the size actually is more relevant for the primary brain tumor as opposed to metastatic disease. So because for primary tumor, early detection actually does make a difference.
Oh, I see.
Right? For metastatic disease, early detection does not make any difference as far as prognosis, as far as the treatment.
Yeah, well, that's again, I think we're developing along the way of early detection, probably not only just size, but also number, but also leptomeningeal disease along the line. Because I think in the future, if we have, my thinking is, if we have a reliable way to predict when the left meningeal disease will start, then I still think giving early whole brain probably will still have a benefit. But for primary, I think I'm a little confused because all the data we have are all brain mets. We kind of excluded the primary. The primary, the problem is we can never really afford screening the patient with every 3 months MRI from the very young age. So cost-wise, that's a
It's
problem.
not possible.
Right. Yes, but the brain mets, we already have the standard care of this frequency.
I have a quick question for Dr. John. So I'm curious that for that like a scalar metastasis, where that pattern is related to like a vascular structure distribution. I mean, if so, we may can think about adding this information into later like training or something.
Yeah, that's a good question because actually Manish Mehta from Miami Cancer Institute, they actually had a poster just 2 weeks ago. They actually are using the so-called black blood sequence to suppress the vasculature. So basically get rid of all the microvasculature and only the brain mets left. So the visually that's much easier to detect. So they actually can identify way more brain mets than what has been identified. But then we were working together to see, even after suppressing that vasculature, using this algorithm, can we still detect more? So again, it's definitely just not a single modality of algorithm. It's gonna be some combination of all the different factors involved. But it will be nice to confirm by the radiomics to see whether the vasculature is a factor. Yes.
I have a question for Dr. Eisenberg or anyone about brain mapping. So for a function like, let's say, motor control of your right hand, like how often is that area different than what like the textbook would predict, like the homunculus in the medical student textbook? You know, is it almost always in the, you know, the left precentral gyrus laterally, or does it differ dramatically from person to person?
So that's a good question. The brain can reorganize its function, and generally speaking, the motor function is pretty discrete. So yes, it's in the precentral gyrus. It's in the middle portion, the hand knob area. There might be some variation in location in pathologic states. It could be in the same gyrus, but also the supplementary or premotor areas may pick up some of that function. And I actually have a longitudinal study where I have preoperative functional mapping, the intraop data, and then post-op scans at 2 months and 6 months. And we're looking at the laterality of the function. So if you have right hand, obviously you're thinking of left brain, and we're seeing that the ipsilateral side will have activations and it can go back and forth. So that's kind of a complicated, not absolute answer specifically, but I don't think there is a specific answer. Generally, yes, we see things where they should be, but in pathologic states, things can move. And it also depends on the duration of the pathology. Low-grade tumors are present for long periods of time, and there's a lot more reorganization that happens. So it's also harder to know, and it's beneficial to do pre-op studies. Where are these functions located? And so I hope that's answering your question adequately.
Yeah, thanks.
So if I may add a sentence, because I think, as Dr. Eisenberg said, those slow progressive disease potentially can cause the distortion of the function area quite a bit, because we see that in AVM case quite often. Because some kids, well, all the AVM they started to have as a baby. So all their lifetime, the brain is rewiring. So we did see a lot of change.
Yeah, that would be different. If
Yeah.
you have something congenital, or something, very young over the years, that the brain, you won't see things in that typical spot. But in someone who was otherwise normal through development, and then developed a pathology, but things are dynamic. And we don't know what the time point is that you may find something. I chose the 2-month and 6-month time points, that made sense based upon the treatment somebody may have and, the surgical care, but those may not be the right time points.
And that's a question I didn't ask you, like, if we have longer follow-up, do you think you're gonna see the pre-op finding dictating how the wire is reconnected?
Well, we didn't talk about this today, but there's brain connectivity, there's functional as well as structural connectivity. And that adds a whole nother dimension to all of this.
For next year?
Yes. So, and I'm looking into that as well, but more to come.
Perfect.
So I have one question actually for Dr. Zhang. You were saying that using the model to predict the lesions in the future, if you can predict in 1 month, that's probably easier than 6 months. If you can predict it 10 years later, someone can have something, that would be great.
So, yeah, but going back to why we started to look how many lesions were missed. Because if we don't see anything missed up to 6 months, maybe there's no radiographic feature there. But since we see so many lesions that were missed within the 6 months period, I would say probably by the time you see a brain mets, even 6 months early, there's probably already a seed there. So, but ideally, yes, if I can predict 10 years later, but it just physiologically, that probably haven't happened.